There are few figures in regenerative medicine whose work has truly reshaped what we believe is possible. Dr. Anthony Atala is one of them. From bioprinting organs to leading groundbreaking efforts in tissue engineering, Anthony Atala has achieved many clinical breakthroughs at the Wake Forest Institute for Regenerative Medicine. Recently, these technologies have reached critical scientific milestones and are bringing real hope in real time to patients facing injury or disease once thought untreatable. Dr. Atala has always been known for challenging the boundaries of science and medicine, but few are aware that in recent years, his work at the Institute has expanded beyond planet Earth and into space, working with NASA and the International Space Station to meet the future medical challenges expected as humans embark in journeys to other planets, including Mars. But what I personally admire most about Tony, who I consider a friend, is his reputation for keeping patients at the center of every discovery and every decision. So now, friends, join me in welcoming into conversation our latest pioneer of hope, Dr. Anthony Atala.
Well, it's so good to see you, Doug, honestly.
See you.
Really. What a treat.
It's been about 10 years, as I remember. Maybe not quite 10 years, because I think that I had my first exposure to regenerative medicine in 2015. And I think you and I talked in 2016. It would have been the year after. So 8 or 9 years.
Amazing. Time flies. Time really flies.
It does. But you don't look any older. How did you do that?
The cells.
That's what everybody tells me. I've been trying to follow Wake Forest, and I was most excited about a recent NSF funding opportunity that came your way, tens of millions of dollars, $100 million or more, is it? Can you tell us a little bit about that? It's so exciting.
Yeah, absolutely. So we're excited about it as well. The NSF came up with this grant call that said, there are a handful of hubs in the U.S., a number of hubs in the U.S., technology hubs like, Silicon Valley in California, or the biotechnology hub in Boston, or the pharmaceutical hub in the Philadelphia, New Jersey corridor. And they said, the NSF, they said, we would like to build more hubs around the nation that are driven by science and innovation. So basically, the whole purpose of the grant was to develop ecosystems and other regional hubs throughout the nation that are driven by use-inspired research and innovation that can drive to new technologies and commercialization so that you can establish more entities in your region and have job creation and upward mobility. And it's a great, great program. It's such a great program and really very visionary from the NSF. Because, if you really think about it, it includes all the elements we all talk about, right? It includes science.
Assisted development has been an issue that W firm has been working on for years now. And I don't know what kind of progress there has been in terms of training next generation scientists and technicians to actually carry through the innovation. Is that part of?
Absolutely. So it has all the elements you think about it really in one grand mechanism, which is science, innovation, workforce development, commercialization, manufacturing, job creation, and upward mobility. I mean, what a great program. It really is a start program from the NSF. We were very excited about the program, and of course we applied to the program based on the ecosystem we had started to develop here. It's a nascent, it was a nascent ecosystem at the time. And we decided this fit our goals as well, what we had been doing here. And they had over 700 applicants, believe it or not. It was a very rigorous process of, basically a year of site visits in a way.
Right.
And then they announced 10, and they announced one in the healthcare space. And it's basically a 10-year grant up to $160 million.
Right.
And they announced 10.
2 years at 30, or is it 2 years at 15?
It's 2 years at 15, and then you keep going back every cycle for more, up to $160 million for 10 years. And it was great. We were felt very lucky to get one of the 10 grants, the only one in the healthcare sector, actually. But really it's all about technology, and trying to bring it forward.
And certainly of all of the, I guess you could say, the skill sets that W Firm have produced through the years, and of all of the innovation, a lot of it has been bringing the technology in to focus with what can happen with it. So I remember talking with you during our last interview. You may not remember it, but I've been watching the footage. So we talked about some of the early regenerative medicine activities that you began to spearhead. And you mentioned specifically, from your background as a urologist, pediatric neurologist, you mentioned specifically using intestinal tissue to try to repair, recreate a functioning bladder. We ran into a difficulty that you were using tissue that was meant to absorb things, not contain things for excretion. And we talked about it a little bit, but as you look back to that part of the conversation, now I know that happened for you somewhere in the low 2000s, right? Like 2004 or 6, somewhere, right?
Right.
So 20 years ago.
Right.
Tell us how far it's actually come.
As it was considered science fiction back then, when we first started the work.
If it was considered science fiction, what did people think of you?
Oh, it's crazy, right? I mean, this was the time, remember, the term regenerative medicine had not been coined.
Right.
Stem cells had not yet been discovered.
Right.
Cloning had not been discovered. So it was very early in the game when we started this work. And, and we really work hard to try to get that first organ implanted in a patient. Up to that point, of course, you had, skin cells that had been placed, but they were partial replacement, just as a biological dressing to protect the patient till they got a real graft from the patient. This was really a permanent replacement that we were putting in. It took a lot of work, a lot of years, and we finally were able to put into the patient. But, if we had known then what we know now, we would have never started with a level 3 organ, because it is the bladder's a complex organ, flat structures being the least complex, like skin, then tubular structures being the second level of complexity. Of course, all these structures have blood vessels and innervation that come in, but definitely when we look at the organizational structure and complexity of the organs, flat structures, things such as skin, they're all complex, right? Even skin's very complex, but architecturally, flat structures are the least complex. Tubular structures like blood vessels or urethras are the second level of complexity. Hollow non-tubular organs like the bladder are the third level. And then solid organs are like the liver, the kidney, the heart are the most complex. And of course, we started with level 3 organ. And if the bladder, we had known back then what we know now, we probably would have never started with a level 3 organ. But by starting with a level 3 organ, it really made it easier for us to go after level 1 and level 2 structures.
Why do you say it made it easier?
Just a lot of the initial challenges, just to get the cells to grow. As 35 years ago, most cells could not be grown or expanded outside the body. I mean, you could retrieve cells, but you could not get them to expand.
Right.
At the time, that was a major challenge for basically most all human primary cells. And so just to learn the cell biology, how do you harvest cells? How do you make sure that you can expand the cells, but that they could retain their function? Just the cell biology alone. In fact, our very first grant, when I, was an NIH grant on cell biology, because, I wanted to figure out how to get these cells to grow. And then, just biomaterial sciences, using biomaterials that you could use that would be biodegradable and non-inflammatory and yet would permit the cells to expand and create structures. It was really uncharted territory in many areas in terms of cell biology, biomaterial sciences. And of course, as we learned that, and, of course, many many investigators that contributed to this worldwide. I mean, biomaterial science has developed and was huge.
It's a huge engineering feat.
A huge engineering. But we certainly learned a lot to get that first tissue created and implanted into a human. And then, and then it made it easier because of the same strategies. We used the same strategies for the other tissues. Really the same, take a biopsy, expand the cells outside the body, use biomaterials. Of course, they were all different biomaterials. And then, how do you shape the structure? How do you make sure it survives? How do you make sure you put it back in? We learned a lot through that process.
So back then, you took a level 3 organ and you worked with it. You learned a lot. That was 20 years ago. Can you bring us ahead and tell us what you're most excited about right now?
Yeah, so from that first organ, we are now at 17 technologies in patients to date. And what's most exciting for us is really every time that we put a new technology into a patient, because that's a whole new area that we're tackling and, a whole new population of patients that can benefit from these technologies. And so, doing things, that we've done in the area of skin and cartilage and, so many different target tissues and organs.
So it sounds like it's pretty exclusively tissue generation in some form that you're making the headway in. When you said you have 17 technologies implanted into patients, are all of those either tissue or complex tissues or organs or?
Yeah, so they're all cell-based for them. They're all cell-based. And then in terms of, or I should say cell/biomaterial based. And we've used cells for therapies in certain indications where you're using cells in a hydrogel or cell in a carrier. We've used engineered tissues where we create the tissue and implant it. So really, and of course we have so many different tools available to us right now. We can make these by hand. We can, print. We can deliver cells with hydrogels. There's so many different strategies now that we can use. And at the end really is how can you make the most impact on a patient's life in the safest manner possible.
We played a little bit of a pre-association game last time we talked. And the goal and the mission has always been the same. You said the very same thing then, and I'm impressed that has not wavered. And on the other hand, time, is ticking by. 20 years is a long time. Is anything close to market? The pat on the head syndrome and it's just as frustrating to doctors as it is to patients, where you're saying, a cure is on the way, maintain hope, right?
Yes.
But the time just goes by. And
well, first of all, there are a couple of things. First is that technologies take years to develop. As you know, the average time to take a drug, a pill, something like, a pill that you take orally, the average time to develop that is, over 15 years, close to 16 years now, at a cost of several $1 billion. So that's just for a pill. And these are much more complex technologies. We're using cells and as personalized medicine and biomaterials. So first of all, it is a long timeline, but certainly it's a timeline that we are used to in the biomedical world, because that's what happens with all the drugs that we take in terms of, timelines. And then the other thing is the ability to...
Can I challenge something there? When you said this is what we're used to in the biotechnology world, does it have to be that way? Has there been enough attention given to new ways to accelerate development? And maybe it doesn't have to take as long or cost as much.
Absolutely. I think, the FDA has really made some strides in that area. Right. They really have, the 21st Century Acts and now newer ways to do things with the FDA, I think that timeline is going to be shortened. I really do.
Are you experiencing that at W Firm, or
yep,
you able to tweak it so that you get things in a little sooner for this? Or that?
Yeah, absolutely. Technologies we're putting in now, we're much more prepared to do those, right? Because we keep doing these, and we have great interactions with the FDA. We consider them really great partners with us. They really want you to succeed,
They do.
They really do.
Yeah.
So I think that's one thing. And safety's paramount. I mean, I get it, and I know why all those rules and regulations are there, and we embrace them because that's what you want to do for the safety of the patient. And the other thing is that remember that even though these technologies, before they get to the marketplace, which they do, before they get to the marketplace, they're still being used in patients. So patients are still benefiting through the clinical trials. Patients are still being treated with these technologies.
Yeah.
Patients are still benefiting from these. And of course, patients are heroes. I consider them heroes when you're first doing this in the very first group of patients. Of course, as you go through the second and third layer of the clinical trials, it's much more readily available and less risky. But still, these patients who participate are heroes in my mind, and they truly deserve a lot of credit.
This episode of the Pioneers of Hope interview series is proudly sponsored by BioXTech Regenerative Therapeutics, a full-service partner for launching innovative regenerative medicine. From donor screening to GMP and CGTP manufacturing, BioXTech provides advanced processing, translational guidance, and global distribution. Their proprietary BioPer technology preserves exosomes, growth factors, and extracellular matrices, unlocking the power of healing at the cellular level. BioXTech, advancing the future of biologics so pioneers everywhere can bring hope to patients worldwide. Check out BioXTech today by scanning the QR code or by navigating to bioxtech.com.
Do you have a story that comes to mind about a patient that you saw in a clinical trial that succeeded, and then maybe a story where the patient didn't succeed, where the treatment didn't succeed? Could you share with us what does a scientist or a physician scientist researcher feel when there's a success and the patient is the hero, and when there's a patient for what are a million reasons are disappointed or less hopeful? We know that both of those exist. Sometimes our trial structures even force them to exist too long.
Right. So one of the things is, I have never really, because I've been involved in developing these technologies, I'm never really directly involved with the clinical trials because, I see that as a potential conflict.
It probably is.
And it doesn't have to be. A lot of people do that. But in my mind, I really keep it separate because I've had a hand in developing the technologies. I don't want to have any bias when the clinical trials are done. So I really don't have. So that's been my rule from day one. But, of course, I've met many of the patients that we have treated.
Yeah.
I've had interactions with them, some that I still stay in touch with them 20 years later.
Right.
Right. And so it's a great thing. And of course, you learn that patients' lives can be changed, but, things can happen along the way, where.
Like what?
Like, for example, you can take a biopsy from a patient's tissue and you can create the construct, have everything ready to go, and all of a sudden there's contamination, right? And then you have to start all over again. You have to start creating the tissue all over again. And that has happened. And of course, that's a disappointment because then you have to reschedule the patient and you have to redo all that. So, the perils of the technology are that they are personalized. So a lot of what we do is we take the cells from the same patient, grow and expand the cells outside the body, create the construct or the therapy, and then put it right back into the patient. Right. And so it's not like things you can take off the shelf sometimes.
It's a one and done. That has an impact on my thinking too. One of the things I've said for several years now is that one of the problems with precision medicine is that it's precision. And there are so many patients that would gladly let good not be the enemy of perfect. They would gladly take an intermediate step to alleviate suffering before that pinpoint accuracy, brings home the bacon. The concept of a broader spectrum of applications on some of the innovation, say that even W Firm has created, that could be applied to numerous conditions. Is there any pathway forward for that, or is that something that, from a physician researcher standpoint, you don't see where that's gonna fly?
Yeah, no, I think you never, the one thing I've always learned in science is you never say never in science, right? And you always leave the doors open to possibilities. So definitely, I think that cell sourcing has been one of the main directives for the field. How do you source cells? How do you use the cells? Can you use one cell for multiple different tissues? So that's a cell type that we have been really exploring for quite a while, and they're very powerful because they're not mesenchymal cells, they're not mesodermal cells, they're actually just like human embryonic and iPS cells. They turn into all three germ layers just like human embryonic iPS cells, which of course those two features are not present in adult stem cells. And more importantly, unlike human embryonic and iPS cells, they don't form the tumors.
That sounds like it could be a candidate for exploring how to have some broader therapies available until the genetic therapies hit, or until the replacement tissues are available.
Yes. Yeah, the nice thing about those cells is that they're, mesenchymal cells, as you know, are very limited in what they can do, right. Or mesodermal cells, very limited. And basically, unless they're iPS cells, induced pluripotent stem cells or human embryonic stem cells, you're looking at, that's the predominant cell out there is mesodermal and mesenchymal cells, which are very limited. And also you cannot really grow them in very large quantities in terms of their expansion capacity. So these cells really are much more powerful in their ability to grow and turn into different things and form different tissues. So it's a cell that we really, but we have been working with that cell extensively.
So can we shift a little bit and talk just for maybe 5 minutes about where you are in your personal / career life? It's almost 10 years since we spoke. How have you developed and grown, and what motivates you to come into the office every day? Can you share just a little bit personally about what goes on in your mind when you're in the car coming to work?
What's always motivated me from day one has been making patients better. As you know, I'm a physician, I'm a practicing physician. I'm still doing surgery. I still see patients every week. And I find that to be so rewarding because I have that one-to-one interaction with patients. Yeah. I can go into the operating room and do surgery and make those patients better surgically as well.
What inspired you to do what you're doing?
I was always inspired since young about being a physician to make patients better. I mean, that's really what inspired me from since the time I was a child. And in fact,
as a kid, you just said, I want to be a doctor to make patients better?
I did early on, and then I changed my mind, and then I went back to it, So when I was really young, I wanted to be a physician. Then I kind of started to change directions, and then I did refocus back on what I really wanted to do. And so I never really intended to do research. I was just going to do clinical work.
Okay.
And then
How did you get bit by that bug?
Well, that's an interesting story. My intent was to, become a physician, get trained, go into practice.
Yeah.
And, when I was in part of my training, I decided, I really like the academic world, but I still didn't want research. I still didn't want to do research. And so at the last stage of my training, my boss-to-be who was Dr. Alan Reddick, who was a surgeon-in-chief at Children's Hospital and Harvard Medical School. He basically called me up and he said, we're changing the training program this year, and we're going to add a research year. And we want you to consider doing that, because this year we're going to take 2 people instead of 1. But I think you ought to be the one that goes in, and I said, well, why? He says, well, because I know you're interested in academics. I said, yeah, but I'm not interested in doing basic research, I said. So I have no interest in that. And he said, really think about it. And I said, okay, I'll think about it. He said, I'll call you in a week. So I talked to my mentors, my professors, my wife. And a week later, he called me and said, well, are you going to take just the clinical track, or are you willing to add the research track as well? And I said, you know what, Dr. Reddick, I've really thought about it. And I've talked to all my mentors and teachers. And you know what, it's still going to be I still only want to do the clinical track. And he said, really? I said, yeah, really. So he said, was your wife home? And I said, yeah. I said, well, can I say hello to her? So he got on the phone with my wife. She got off the phone, looked at me and said, you got to do that. You got to do that research track. And I said, why? I said, well, he thinks that would be good for you, so you ought to do it. So I said, okay. So that's how I ended up, doing research, being exposed to it. And that really, turned a new page for me.
Do you like the research now more than the clinical work? If you have a preference?
I like both the research and the clinical work equally. I really do, because I feel that one feeds the other, right? The fact that I can't have what I need necessarily to take care of my patients the way I would want, because, right, there's nothing more devastating for a surgeon than to be in the operating room and know that you're fixing this patient with something is suboptimal, right? It's not the patient's own tissue. You're putting a piece of metal or a piece of plastic, and it's not the best option, but that's the only option you have. And so it inspires me to keep moving forward in this area. It really does.
So what's next? If I come back in another 10 years and, gained a little weight, lost a little hair, I don't know how that goes, right? Where do you want to be 10 years from now?
So, a lot of what we've been doing in the last few years is really making sure that we can increase the availability of these tissues and organs for patients by making it easier through manufacturing. So we've gotten, not just are we doing the innovation, but the problem is that we're very, in the U.S., we're very good at innovating in research. But then you innovate in research, and then when it gets time to manufacture it for a product, you have to start all over again.
Why?
Because now you have to take whatever you did for research and make it into a standard operating procedure. It's going to be a process development. It's going to be a manufacturing process. And it's totally different, right? It uses different agents, is qualified materials, release criteria, everything that goes along with it.
Yeah.
So the question that we asked ourselves a few years ago was, why can't we innovate in manufacturing at the same time that you innovate in research? And of course, that was really, a precursor when we started doing the 3D printing work. But we really then got into standards and, what are the things that we really need to do to make sure that it makes it easier for everyone to manufacture. So manufacturing has been a big area for us in terms of making sure that we get the standards for manufacturing.
Has that worked for you?
It has. We launched a number of projects that anyone can benefit in the field. Working on a universal media that anyone could use right off the shelf, so you don't have to go through that process and qualify your media. A universal bioink for 3D printing, bioreactors that you can use for not just for growing the tissue, but for transportation, manufacturing know-how, and also the next generation, in space manufacturing, doing things for our space travelers and with NASA and ISSNL, the National Laboratory for the International Space Station, and with multiple partners that we have. And that's really a new frontier for us, literally, to make sure that we can get those tissues and have them available.
In general, what's the superiority of tissues manufactured in space versus in gravity?
Well, we're two things about our work with the space program and ISSNL and NASA. One of them is that what we are doing in space is helping us in terms of our manufacturing technologies right here on Earth. Right. That are going to benefit patients today as we develop these technologies. And the second is for us to really think about, the future missions of like the mission to Mars, where it takes 6 months to get there, and to make sure that we have the ability that if our space travelers get in trouble, that they have something up in their station that can help them out. So there's so many different areas. Those are just two of the things that are, advantageous for us to do this work. And of course, you have to do the work now for the future.
And so are you actually talking about helping people on their way to Mars? Is that an active conversation here at WFM?
Yeah, so the conversation that we have is really, how can we manufacture these tissues, and how can we improve manufacturing with low gravity conditions? Okay, that'll help us here and in space. For us, any time that a technology gets to a patient, that's a win. So for me is keep working on all these technologies to keep advancing them. Okay. And the goal is not just to have more technologies. The goal is to have more patients that can be impacted positively and can benefit from these technologies.
Amen to that. As you know, I usually ask if the guests have the question they'd like to ask me.
You've done so many things in the field for so long, and you've been such a, great contributor in so many different aspects. What's next for you?
What I'm most excited about is that we developed a cell-based therapy that the early results are showing that it has an impact on restoring vision in people with geographic atrophy.
Well, that's impressive.
We dosed our first patient. They've gone through one treatment, I think, and 8 or 12 weeks out now, still monitoring the safety. And they had an acuity change of 2200 to 2080.
Wow.
And
wow.
Imagery that shows the reduction in GA lesions. It's really real. So
wow, that is great. Well, I'm looking forward to seeing that.
Yeah. We'll just see where it goes.
That's great. That's admirable. Thank you for what you're doing.
Thank you very much.
Great to see you again.
To survive, faith, hope, love.